That'd be so hard to hear. Like the day after that, they had left like the windows open in here, so it was nice and cool. That one day, that was the day it was like ice outside. And all that. So that one day we got some relief. It's not gonna help really if I open them today. All right, guys, I'm gonna go ahead and start it. Of course, if you guys do want to join like after it gets started, or if you get kicked out or something, that code will appear at the bottom of the screen, like every other slide or something like that. So FYI. Alrighty. And I'll post this Kahoot online as well if you guys want to practice through it too. All right, so first things we're going to talk about are safety rules. You will get quizzed a little bit on these, just kind of obvious ones, I feel like, but yeah. Right, where should broken glass be disposed? Right, of course, sharps container. And keep in mind, even if it is contaminated glass and it has like bacteria on it, technically sharps container is also a bio container. So, all right. All right, moving on to the stains. Okay, for the stains, you're going to want to know the stains in order, right? So, and I think I list all of them at the end of when we go through some of the questions for them here in a moment. But what stains you would be using in order. The steps, I don't care about how long they sit on there. I don't care about water rinses or anything like that, okay? But I do want you to be aware of those steps, each stain and what its purpose is. So yes, the stain itself, if we're doing like endospore stain, obviously it's stained for endospore, but can you tell me what malachite green does in endospore stain? That's the kind of question you're going to get asked on the exam. I can see that on the screen clearer, but I'm like old, so I need like reading glasses. So what a positive and a negative look like for each type of stain? Like if it's positive, what color would it be? If it's negative, what color would it be? Or would it have a color at all? And identify each in a mixed sample. If I were to give you like a picture of like, and I'm just gonna be honest with you, I'm not gonna give you a mixed image in a microscope and then be like, you know, I don't even know what I would ask about it. I'm usually just gonna give you a population, positive or negative. But if I could have given you a mixed, be able to tell them apart, right? If you can know them that well, then you're in good shape. What happens if you skip a step? This is especially true, of course, I think I've already warned you guys about this, the gram stain. Be aware of what happens if you miss, you know, the alcohol step in the gram stain or something like that, right? So if you can walk yourself through the ideology of what it should look like at each step, because you know what's going on for each step, then you should be doing pretty good for that. All right, so let's get into the questions for it. All right, there could be more than one for this. So what is the purpose of heat fixing? What does the heat fixing itself do? Let's say that. Yeah, so good job, guys. This is going to, in fact, be killing the organism, which we know that's definitely going to do that because it's basically super, super hot. And then adhering it to the slide so that whenever you are doing your rinses, it doesn't rinse off. There's one other thing that it does, which is help preserve like the structures of the cell. Just if that is another thing that it does. But I feel like these choices are kind of clear. And on the exam, since it's Scantron, you won't have to pick like multiple. It will only ever be one answer. So that's me being lazy. Scantron can't tell whether you choose multiple things. So I didn't want to read people's handwriting, and that opened the door for handwriting reading, and I didn't want to do it. So what is the counter stain used in the gram stain? The counter stain. All right, so our counter stain for our gram stain, you guys must have got this right, that's safranin. So keep in mind when we do our stains, the first stain that you're using is typically going to be your primary stain. Primary first, so they go hand in hand. But the primary stain is also going to usually be what is going to give you like the main answer for your question as well. So you usually always have that primary stain, and then there might be other things in the middle like decolorizers and stuff like that. But typically most of our like differential stains, which is what these are, to help us tell the difference between whether they have a structure or not, we'll have a counter stain at the end as well, just to help visualize typically the negative that got decolorized. So in this case, we're decolorizing the negative literally and staining it pink or red. All right. What color will a gram negative bacteria be after the gram stain? Right. So this is going to be red or pink, but I'm always going to, in my test, call it red, just so you guys know, because I think it's easier to remember the red versus the purple thing. So, yeah. Safranin. So what is the first stain used in the capsule stain? If you guys remember this one. Okay, so the first stain that we use in the capsule stain, and believe me, I'm not judging you guys because I know we're just getting ready to start studying for this, but the first stain used in the capsule stain is going to be Congo red. That's when we put the drops and then mix our stuff in and then spread it out like a blood smear. That's the capsule stain. So that's Congo red, and it is a negative stain. That's probably the next question. Watch it be. Oh yeah, what type of stain is Congo red? So this kind of has two answers technically. Yep, good job, guys. So you remember this is a negative stain, and negative stains are staining the background. So good job. That's correct. I know. It's funny because like people often say that to me. They're like, well, if the test was like this, I was like, to me it is, but I think it's the format. Sometimes it stresses people out more than anything. And like having the papers and like, I don't know, it's just a stress. Yeah, exactly. Hi, welcome to the test, guys. Yeah, okay. So this is the pure culture. What stain is pictured? All right, so that is the endospore stain. A lot of these stains I will have like a microscope up and it'll literally just say like, what stain is this? Or what is this stain, testing for the presence of? It's pretty easy to pick out most of these guys. When you see endospore stain and you've seen a couple of pictures of it and you remind yourself that's what it's supposed to look like. Or the capsule stain, which everybody knows, you'll see like the halos in the background will be colored, that sort of stuff. So it should, when you see them, be relatively simple. And if you have like questions about it that doesn't, like if you have a question like, is this pink or purple? Or something like that, like is this red or purple? I can't really tell. Stuff like that is okay to ask, because I actually had colorblind people come through the course. They literally can't see the color differences. So I'll have to come and explain it to them. If I can explain it to them, then I have to be able to, you guys can't tell if it's purple or pink. Sometimes it's hard, especially when you're stressed out and you're like, is this really purple or is this pink? Because sometimes that red, for whatever reason, didn't rinse quite right and it leans a little purple. And you're like, is this supposed to be purple? So it's okay to ask. And I'll tell you like, oh, that's meant to be red. you know, and then you complete your correct answer. It's okay to ask questions, okay? You won't get in trouble or get points off or anything like that. Anyways, that was endospore. We saw the little green bubbles that look kind of bluish green in the picture. And then the cells themselves, the vegetative portions, are stained red with the safranin as the counter stain. Alrighty, multi-select. What is the term for the red structures in this image? I always say it before we go into it, but if you weren't paying attention, you get to decide. All right, so the red structures, those are the cells that are making the endospores, or that are the type of cells that could make the endospore. So those are vegetative cells. Remember, endospores are completely dormant. They don't do anything. They don't move. They don't do anything. They're literally just entirely dormant, waiting to be woken up again. So the difference there. All right. Which of the following bacteria would be acid-fast positive? All right, so this is going to be Mycobacterium flea. So that is Mycobacterium. Anything will fall into that category, Mycobacterium specifically. So don't get tripped up by Mycoplasma or Micrococcus. I won't get into those, but those aren't going to be involved in that. Now, for the endospores, remember, that's going to be Clostridium and Bacillus. Okay, so there's two for that one, but there's really ones. Really freaking hot here. What is the primary stain used in the acid-fast stain? Okay, yeah, so the primary stain for acid-fast is going to be carbofuchsin. Was the majority pick, at least, so there's that. That is the one, that carbo group is what allows it to interact with the waxiness of the glycolic acid on those Mycobacterium surfaces. Alrighty. In which staining procedure did we use steam? All right, so this one is, yeah, it's okay. So this is the endospore one. Endospore, remember, because they're dormant and they're those hardy structures that can pretty much resist anything, it's hard to get the stain to penetrate those endospores. So the steam helps the malachite green penetrate into those. All right. Which of the following structures allow for bacterial motility? Yeah, good job. You guys got it. So that's going to be flagella. All right, so that's it for our practice questions for the stain stuff. So remember, we learned about the following stains. Simple stain, so don't forget about simple stain. It's literally just using one stain color to stain the cells of color. It gives us some idea of contrast of what's going on, instead of like, remember when we looked at the motility stuff and it was so hard to get like the light just right, just to see the cells even at all. So the simple stain allows you to at least see that a little bit better. Then we have the gram stain for whether it's gram positive or negative, acid fast for whether or not it has that mycolic acid waxy covering. The capsule stain, whether it has that capsule. Remember that capsules are going to allow for adhesion to surfaces as well as evasion of phagocytosis, getting eaten up by your white cells. And then the wet mount or the motility one that we looked at where, you know, was literally them swimming around. We talked about the flagella with these questions, but and I'm not going to have you look at those in a microscope, just FYI. you get questions about it, they'll just be like a question about it. And then endospore, that's of course going to be those protective structures that they form in harsh conditions. And they'll go completely dormant. And I'd be aware of like the terms involved in that cycle. But all right, moving on to ELISA. I say know this test inside and out. Definitely what I'm saying here is understand the mechanisms for it. Right? How does it operate? Why does it give positive? Why does it give negative? And this sort of stuff. So first of all, my favorite question for the test typically is, what does the E stand for? All right. So that is enzyme. Great job, guys. Pay attention. Enzyme linked immunosorbent assay. All right. What does an indirect ELISA look for? What is it looking for in the patient? Alrighty. Good job, guys. This is, in fact, we're looking to see if the patient has the antibody. So remember, if that patient has been exposed to this, pathogen at all, they may not still have a present infection, but they could still have antibody lingering from having had the infection. And even that may give you some information you need medically when you're diagnosing something. All right. Oh, that's it for ELISA, I guess. The principles of how the assay works, be aware of how what the antibodies are interacting with, right? Antibodies, they interact with antigens, right? It's a very specific interaction there. Be aware of what antibodies and antigens are. They're both, well, antibodies are always proteins, and antigens, anything that your body can see as foreign. And antibodies will react specifically to an antigen, and there's usually no cross-reacting between them. So it can be very specific on how they can use them to diagnose things. Indirect versus direct, we just said indirect, which is like the ones that we actually practiced in class. Those ones we're looking for antibody in the patients, as opposed to the direct, where we had that sandwich looking for the presence of the antigen in the middle, right? And then we had color changing, how that happens. The enzyme cleaves the substrate, right? Enzymes always act on substrates. In this case, the enzyme that's on our detector antibody will cleave on to the substrate when we add it in, if it is present there, and then cause the color change. If it's positive, right? If it's negative, no color change. And then rapid testing. We mentioned a little bit about rapid testing with the how this applies to COVID home tests. So we'll have antibodies there and already implanted in there. They've got their own like enzyme there. If your sample runs through on that strip and contacts it, it will cause a color change and create a colored line there. Of course, all you see is the stuff moving through and then a color change or not, but that's what will be going on there. Similar to what we see in a pregnancy test. So, all right, then the DNA stuff, the stuff you have to know that we didn't practice in lab, but I gave you that study sheet for, and like I said, most of that was covered on the test too, so it shouldn't be news really. And it includes transformations, that transformation reaction. So what does PCR do? All right, so yes, it's going to be amplifying your DNA samples. I was about to give you away the letters of that. I'd imagine that's what my next question is going to be. I always get mixed up when the questions are coming. Which DNA fragment is largest? This is a common question on the test. Won't be these exact numbers necessarily, but be able to recognize which one would be the largest of this group. All right, good job, guys. That is in fact going to be 35 kilobase pairs, so kilo being 1,000. And I tell some people if they have problems remembering how to tell that, just write them out fully if you need to, and then compare them. It's okay to do it. All right, which of the following DNA fragments will travel the farthest on an agarose gel? Okay, so on this one, it's going to be the 100 base pairs, and you guys pretty much got that right. But keep in mind that the reason for that is that the smallest ones are going to be able to travel faster through the gel because the bigger ones get caught up, kind of like you can imagine they would be caught up. It's easier for the small guys to get through. What type of cells are capable of taking up exogenous DNA? Right, the term for these guys is competent. It just so happened that the example that we were going through in class involved E. coli. And you guys didn't choose that one, so that's good. But yes, so not all E. coli are competent, right? And not all cells are in general. So competent is just the term that meaning they can take up exogenous DNA, capable of doing that. All right, so for the DNA stuff, be aware of DNA structure and function. I'm not going to ask you about like replication and stuff in here. That was lecture stuff. So that's not going to happen. But when I'm talking about like PCR and how it works, if I ask you a question about that, it's just a good idea to know what a nucleotide is, maybe like that sort of stuff. DNA fragment sizes, we just already talked about that. Agarose gel electrophoresis. We had in the exam, there was a question about like what direction like DNA will move in the electrophoresis, and that's going to be towards the positive end because DNA has that negative charge from the phosphate groups in the backbone, right? Okay, so be aware of that as well. How PCR works in a practical sense, what we would actually be using it for to test, how we would use it for to test, because remember, just because I swab your nose and you get some virus out of it, doesn't mean that there's enough genetic material for me to actually do any realistic testing on. The PCR isn't always doing the testing. It can sense the presence or absence of something, but it can't, you have to have, first of all, you have to have the sample and you have to have the primers to test for the thing. And if you're trying to do something else with it beyond that, like putting new genes into a plasmid or something to make, human insulin in bacteria, then you need to have enough DNA to work with for that. So that's kind of can be the point of it, other than diagnostic, which is another purpose. Remember, PCR stands for polymerase chain reaction. Those are not the steps involved in amplifying the DNA. That's just the name of the procedure. The steps involved are going to be denature, anneal, and elongate. Okay, so denature is just breaking apart the strand. Anneal, we're sticking on the primers that you have to build off of, because polymerase needs that. And then elongate is just elongating, adding the nucleotides in the 5' to 3' direction. Okay. Bacterial transformation steps and the results for each media type. So this didn't ask any questions about any of that. But if you guys can remember, we had the 3 media types. We had the regular nutrient agar, we had the ampicillin one, and we had the ampicillin with the arabinose, right? You remember the purpose of those and what is being triggered to be made by the bacteria in each one, and what kind of bacteria, whether they have the plasmid or not really, is what that's talking about, would be growing on each one and why. Then you're probably in pretty good shape for that. Okay. Even if you need to imagine acting it out at home or whatever it is to help you guys study for that, it's fine. Or you can build your own silly little get up like I had. Okay, and then the plasmids in general. Okay, that's a very commonly missed question on the exam, like what type of biomolecule are plasmids? And I'll say it in the class, and then people will sometimes get it during the lab, sometimes not get it during the lab. And then I'll reiterate, yeah, plasmids, y'all, they're DNA. And then everybody will answer protein on the test. So let's just briefly remind everybody. DNA is, it's comprised of nucleotides, we know that, but it encodes for with those triplet sequences, codons on mRNA that then translate into protein, right? So DNA codes for protein, basically. So whenever I'm talking to you guys about the plasmid and like the arabinose gene products and the GFP product and everything like that, that's made from our plasmid in that P-glow plasmid in that example we had. Anyways, they're making proteins, but the plasmid itself is DNA that codes for the protein. So the plasmid is DNA, right? It has genes that code for the proteins. So don't get that too confused. I don't know where the disconnect happens, but somewhere it does, because that's usually the popular answer. So DNA, there's circular DNA. Oh, the eukaryotes. I hate them, y'all. And they're going to be on the midterm. They won't be on the final. I'm just letting you guys know. But yes, this is more or less just being able to recognize pictures, basically based on differences. And it might ask some questions about some of the life cycles that it talks about in the worksheet. So which organism is this? All right, so this one is penicillium. So it was that pink and it looked like it had like the paintbrush structures on it. This is definitely ones that you just have to go back through and remind yourself what they look like. All right, what is the name of this organism? This one. This one we are not. This is the one I told you I'm not going to put on the test, but hey, there it is. On the practice. So every semester previous to this one, I put Giardia on the exam. But I felt so bad about it in the past because it's so hard to just see it and find it, just everything. So now I don't do that anymore. But that is what it would look like. And that clearly was not a picture taken from one of the slides that we used in class. So that was one from online. So that's, yeah, that's cheating. I feel like I put that on there. All right. So be familiar with the organisms that you had to draw and the important structural and reproductive differences. If there are any that are obviously different, be aware of those, okay? The concept of the trophozoite versus the cyst. Remember the trophozoite, troph being like eat. Like we talked about autotroph, we talked about heterotroph. Troph, like a trough, eating, okay? So if they're eating, that's the living, free living form of it, right? The cyst is like the endospore. So it is completely dormant. It is not going to be doing anything, right? And then we have identifying based on differences. I mean, I feel like the aspergillus versus the penicillium are probably the two that I'd be the most familiar with. It would kind of be like the question we just saw previously with this penicillium. And then I will have at least one other protozoan that you'll have to identify. I think I mentioned to you guys the most likely scenario is going to be that it's going to be trichomonas. I'm just saying, because it's easy to find, it's easy to see. It has an identifiable shape. It's got like you can see the flagella on it in the microscope, and you can even tell when you look at the microscope that it'll be on like 40 instead of like 100 or something like that. So it's easy to tell that it is a eukaryote. Yeah, FYI. So FYI to anybody that's watching this when it is posted online as well. You know, trichomonas. It has that pink color, I'm pretty sure. That's identifiable as well. Okay, moving on. General bacteria growth. Like just looking at them growing in general. Here, okay, well, whatever. We'll get back to that. Which is not a term used to describe colony morphology. So yeah, we're coming all the way back to that, and you're gonna be at least familiar with what term is associated with colony morphology or cell shape or whatever. So we're looking for one that is not used for colony morphology. So the word one for this one, a little bit tricky, hyphae. Hyphae has to deal with basically mold structure just itself, but not colony morphology. The colony morphology has to deal with the shape of like the colonies on the, literally just describing them on the media. So it's just a matter of knowing the terms. What is the name of this type of culture? It's hard to tell. I had some up here just a second ago. I don't like the picture of it. If I were to ask you about this, I'll probably have them in a rack. That's where you can actually see it and interact with it. But yeah, that's a slant. It just was held at a weird angle. It looked like it could have been anything, honestly. But yeah, if I have that question again, it'll be like literally a tube with a slant in it. You can see like, okay, this is obviously, oh no, I'm not meant to trick you guys. You're not gonna have weird pictures like that. All right. Next, how was this organism isolated in this picture? What is the technique used for this one? Yeah, streak plate, I'm not gonna lie, it's gonna appear on both the midterm and the final. It's an easy one, so at least there's that, but be aware of like what it looks like clearly and what it's used for, isolating colonies, right? Which arrangements of bacteria do you observe? Okay, good job. So this is going to be strepto because we had mostly chains in this. You could have also picked out a diplo if you really wanted to, because yeah, when we look at arrangements, we're trying to look at all of them that are present in your viewing. On the exam, when I ask you this question about what's the arrangement of the bacteria, it's just going to be one term that I'm looking for. You're going to do the one that best fits whatever you're looking at. Okay. Yeah. What is the name of the shape of these bacteria? All right, so these were spirillum. They have that sort of S shape to them. Of course, we didn't work with any bacteria that had this shape in our lab, so you're not going to see that in the microscope. Go figure. It's either going to be cocci or bacilli. Just heads up, okay? So those would be your choices. Of course, there are some people even have some in their own samples that are coccobacilli that are kind of like that oval shape. But I won't be putting that on the exam. That's kind of tricky. That's tricky business. I'm not looking for that. All right, so for the general bacterial growth stuff, colony morphology, beware of the terms. I don't need you to get too specific about what each term means, because probably on the test, what I'm going to ask is what we had earlier, like which of the following is a term for colony morphology. That's more likely what I'm going to ask you. So I don't need you to tell me what umbonate means. I'm not that concerned with it. If you can recognize that umbonate is related to colony morphology and not cell shape or arrangement, which is usually what I'll compare it to, then you're probably doing all right there, right? Media types. Here we're talking about maybe general growth media versus selective and differential. We talk about that more in class, but just be aware of the fact that there are types of media that maybe some bacteria won't grow on as well as others. If it has antibiotics in it, for example, right? So we're not going to get into that too much. And that also is talking about like slants, broths, plates, and what agar is and where it came from. Okay, agar we know is a solidifying agent that came from seaweed. Okay, so be aware of that. And I won't ask you about specific kinds of selective or differential media yet. That's on the next test. We're learning about that in our next lab, our main lab. So be aware of that. Then we have the isolation techniques. Streak plate is the A number one. You want to get individual colonies, because if you select an individual colony, you can start a pure culture with that individual colony. A colony comes from the growth of one cell. So it's one cell multiplying up on itself. Cell shapes, we already said bacilli and coccus are the main ones that you want to be aware of. And then cell arrangements, that's mostly going to be staphylo and strepto. Those are the two that I would be aware of. I'm not going to pop you out an irregular, like a no arrangement situation. It's going to be either staphylo or strepto. Okay. Those are two I want you to be familiar with. Okay, into the microscope stuff and how to use them properly. If you observe a specimen at 40x objective and have a 10x ocular lens, what is the total magnification of the specimen? Okay, so this is going to be 400x. For those of you who don't know, you just multiply them. So that's that. Which objective lens do we use for oil immersion? Yep, that's going to be 100x. It should be only 100x and always 100x. Alrighty, so with the microscopes, be able to calculate magnification like we just did. It's going to be multiplying, understanding the operation of it, know all the parts. So this didn't really test on any of the parts, but I mean, I feel like at this point, if I were to put a piece of tape on a piece of the microscope and give you some choices, maybe 4, maybe 5 tops choices, and have you guys select what part of the microscope that was, you probably would be able to figure it out. If you are unsure what a condenser is, or what the difference is between the objective and the ocular lenses, or what a nose piece is, then those, maybe look up those, but don't waste your time too much studying stuff you already know. How to use all of the parts. I mean, I don't know what to tell you on that. Understand why we use oil immersion. That's going to be to reduce light scatter. Right? That's the goal. Reduce light scatter, and that increases the clarity of the image. Okay, I forget about Kirby-Bauer every time, y'all. Okay, Kirby-Bauer, antibiotic susceptibility and resistance. Which antibiotic is the bacterium least susceptible to in this? All right, good job. So that is correct. It's going to be E, because that's the one where we had it wasn't affected at all by the antibiotic, right? So it's absolutely not susceptible to it. It used to be an extra credit question. Okay, so Kirby-Bauer, know what a zone of inhibition is. That's just the clearing around the disc, yeah? And how would you measure it? Completely all the way across, right? It's not the radius, so it's not halfway. The minimum inhibitory concentration, I don't need you to calculate it or anything like that. Just be aware that the minimum inhibitory concentration, or the minimum amount of antibiotic to affect the bacteria that you're testing with, is going to be at the edge of that zone of inhibition. Right, that's the lowest concentration it'll be at that it's effective. The Mueller-Hinton auger, what its purpose is and why it is the way it is. It is going to be a very defined depth, as well as have pH buffering to allow the antibiotic itself to be able to function properly, not be affected by pH, as well as diffuse evenly throughout the media. It's got to be, comparable between plates as well. Okay. And then we have bactericidal versus bacteriostatic. I'm sure most of us are familiar with these terms by now. Bactericidal, killing the bacteria. Bacteriostatic, going to be that it's just inhibiting its growth. And we looked into how that works and how we tested for that by testing in that clearing area, that ZOI, for our own stuff. Okay. All right, that is everything that I have for review for the lab stuff. Obviously, if you have any questions of the material, please don't hesitate to contact me. And remember to take study breaks. Don't cram like consistently. Take an hour, cram, and then take a break, and then cram some more. Take some breaks, y'all. I know you guys like to cram. I know that's your thing. So do what you want to do. I will say that while you guys are here, let me stop recording this because other people don't need to hear about